Good day. Now I'll be discussing to you about the proteins. So first, let me define you what is proteins. So proteins are naturally occurring unbranched polymers in which the monomer units are amino acids. So I'll be discussing to you about the amino acids because the amino acids are the building blocks of protein. But first, before I discuss to you the amino acids, let me give you the beneficial factors that a protein functions in our body or in every organism. So first, it functions for the synthesis of enzymes, certain hormones, and blood components. So when we say synthesis, it is the formation. So enzymes are protein. And another example for proteins are the hemoglobins, which is a blood component. The myoglobin, which is a protein which will be found on the muscles, because from the word itself, myo means muscle, and there are also other hormones that is a protein. So that's it. Another is that it helps in the maintenance and repair of existing tissues. Then if it can maintain and repair existing tissues, it can also form another tissue or new tissues. And lastly, it could also be used for energy. So that's for the functions. Now let's move on to the characteristics of protein. So usually, I will impose a question to you guys. So, what distinguishes protein from the carbohydrates and lipids? So, what distinguishes proteins from the others is that protein contains elements such as the carbon, oxygen, hydrogen, but also it contains nitrogen. So carbohydrates and lipids do not contain elements such as nitrogen, usually contain nitrogen, but here in the proteins it has nitrogen, and also it has sometimes sulfur. It contains sulfur element, and for certain special proteins it contains elements like phosphorus and iron. So one example of a protein is the casein. It is the protein of milk. Then another is the hemoglobin, which is the oxygen transporting proteins of blood. Now let's talk about the amino acids and the building blocks for protein. So amino acid it is an organic compound that contains both an amino group and the carboxyl group. So amino acids found in protein are always I know how to read this. So these are the amino acids that, oh my god, how do I write this? Anyway, is the amino acids. So what does this mean? I will just explain this to you. So it is the amino acid in which the amino group and the carboxyl group are attached to a carbon atom or carbon 1. So in protein, the main building blocks are this. So I have drawn first the structure of the building blocks for amino acids. So to continue, so it will start first in the carbon 1 or the carbon atom. Then attached to it is the hydrogen. Now, it's lacking of one. Take note that carbon can attach or has arms, 4 arms. If you could see this and this, it has 4 arms. So, on its side here, it has carbon number 2. So above it is the oxygen, 1 oxygen, and attached to the oxygen is the hydrogen. Now, the carbon number 2 has double bonds with the oxygen. So this group, which I had just encircled a while ago, is the carbonyl group. So this is the first, this is the first side of the amino acid, and the second one is this, the amine or the amino group. So in amino group, other arms of carbon bonds with the nitrogen. Above it is the hydrogen, and below is another hydrogen, which makes it 2 hydrogen being attached to the nitrogen. So this is the amine group. Now if you would write it, well, so this is the what I was talking to you about, this word, the amino acid, in which the amino group and carbonyl group are attached to the carbon atoms. So that's for oh, I forgot. Here was placed below, but here it was placed above. So what is R? So R is the side chain. The R group is the side chain. So this is the R group is responsible for the polarity of the amino acids. So it distinguishes the amino acids from the others, both physically and chemically. Now we have the so-called standard amino acids, and we have 20 standard amino acids. So standard amino acids are the proteins, are the amino acids usually found or commonly found in proteins. So amino acids are grouped according to the side chain polarity. So R group is the responsible for it. Now we have 4 categories. We have the non-polar amino acids, we have the polar basic acid, we have the polar neutral, and the polar acidic amino acids. So another topic that we'll be discussing about proteins is the uh, protein denaturation. So when we say protein denaturation, is the partial or complete disorganization of protein characteristics due to the disruption of its structure. So we have um, four structures of protein. First is the primary. So when we say primary, it is usually straight, or its molecules are so primary is just like this. The molecules are, or the structure is like this. Then the the secondary, it's like a helix for the secondary. The tertiary is now a lot more, so it binds. It usually tertiary structures are the structures are, there are certain, because of the polarity, some binds or attaches to its the R group are being binded, so it makes the structure also binded because the R group attaches to another to add another. And lastly is the quaternary. So this is the complex type of structure because if we example thread a thread that is not being um, organized well, so it will look like this. So let me give you an example of the protein denaturation. So take for example we have an egg. We will cook an egg. So the main triggering factors in protein denaturation is actually heat, because heat cannot be a catalyst in the chemical reaction of our body. So when the egg is heated, if it is being boiled, the this the structures of the protein, may it be primary, secondary, tertiary, or quaternary, it is being disrupted. So the natural quality of a protein is being damaged or being changed. So that's a good example of protein denaturation. Now let's talk about the protein hydrolysis. When we say protein hydrolysis, it's all the peptide bonds are broken and freeing all the constituent amino acids. So the products the only product that's being formed as a result are the amino acids itself, since protein are blocks of amino acids. So what do we mean when we say protein hydrolysis? So in hydrolysis, it is the removal of hydrogen and oxygen. And hydrolysis is the opposite of hydration. So during hydrolysis, it needs an enzymatic action. So that's for the hydrolysis. Now let's move on to, every protein has its shape and every shape has its function. So there are three shapes of proteins. We have the fibrous proteins, or those proteins that is longer than the others. So example for these are the keratin, the collagen, which is usually found in our skin, hair, nails. Another one is the globular proteins, which the shape of it is like a helix. And the third one is the intermediate proteins. So intermediate proteins, or the membrane protein, are the proteins found in the membrane of the cells. And then